As this shows, this is going to be a joint presentation between Poppi and myself.
Should I introduce Dennis
No,
now?
I wouldn't bother. The reason we're presenting this is we've come back to Brisbane about the same time in the early 1990s. Neither of us had any research funds, so we thought if we pooled all our funds, we'd have twice as much as nothing. So what we're going to do today is go through a bit of a transition where we look at, I'll do some industry background, look at some problem definition, talk about some of the applied solutions, and then Poppi's going to just dazzle you with some science around the underlying concepts. So just to give you a bit of background for those of you that probably aren't as aware of the significance of the Australian and Queensland beef industry, Australia has about 3% of the total cattle population, but we're the third largest exporter of beef. So we're a big player in the total scheme of things, and about 2/3 of our beef's exported. So the export market is pretty important. I'm going to be talking largely, or we're both going to be talking largely about Northern Australia, the part in grey. And this has an industry that's worth about $5 billion a year. About 10% of that's in the live cattle exports, which go to Northern Asia and Northern Africa. Most of the industry is based around a fairly extensive production system. Low inputs, low outputs. But there is some intensive production around the coast in the higher rainfall, even some irrigated agriculture, and certainly some feedlots in the southern part of the state. And you might have heard talk recently about mosaic-style irrigated agriculture up in the Gulf, using some of the water that would otherwise go out into the Gulf. But one of the features at the moment about the northern industry is the low profitability, largely based on a high debt structure, and also the fact that costs are increasing much more quickly than the prices of beef. So talking about just the growth of cattle, and I apologise to those of you that are familiar with this, but really we're looking at a system here where on the horizontal axis we've got time or age of the animals, and on the vertical axis the live weight. And this is a pretty typical production system in low quality native tropical pastures, where we get good gains during the wet season, we lose some of those in the dry, and so on as we go across a series of years and dry and wet seasons. So we end up with what we call this sawtooth pattern of production, which means that cattle are not killed until about 3.5 years of age or older in a lot of cases, and quality is low accordingly. And the problem with this, of course, is that if you look at this graph here, we've got, once again, age in years, either live weight or carcass weight on the vertical axes. And these are some of our major markets, which are delineated by the age of the animal and the live weight range that's acceptable within those. And you can see that if we follow this sawtooth pattern of growth on the low quality native pastures, we miss a lot of these markets here. Usually end up in the USA hamburger market, but we miss some of the higher markets. Even with the higher quality native pastures, we hit some of those, but really to push up into these higher value and higher quality markets, we need to increase production along the lines of what's shown here, where we're keeping the animals in a higher plane of nutrition virtually all the way through. And this means that we can kill the animals at a younger age, and that has implications for quality. So if we finish even 12 months earlier than currently, it has major implications to the quality of the animals or quality of the meat that we're selling. And it means that we can often hit some of these markets or the grading systems like MSA. Now MSA you've probably seen when you're shopping in Woolworths. It's a consumer guarantee grading system where we can give some guarantee to the consumer of the quality of the meat that they're buying. And it's based on things like the breed, the age of the carcass, fat cover and marbling and so forth. And there's usually a premium, fairly small premium in cents per kilogram paid for that. There's a new scheme now, certified pasture fed, which means that if you hit this MSA grading system, if you're compliant with that, and most of the production's been on pasture, then you get an extra, well, talk is of an extra 40 to 50 cents a kilogram. So when you add that up, if you've got 40 or say 60 cents a kilogram over a 300 kilogram carcass, that's an appreciable addition to the value of that meat. So what we're intending, really looking at doing, is going from the veritable sow's ear in a low quality tropical native pasture and trying to produce something like this highly marbled piece of meat. The reason that we're getting this sawtooth pattern of change in growth is largely because it's reflecting the quality of the diet that the animals are consuming across seasons. So if we look across years once again here, across seasons going from dry into wet and so forth, this is the quality of the diet from some of our own studies, looking at crude protein content and digestibility of the diet as indicated by fecal NIRS, which is just a method that we have which has been developed in the last decade or so, which allows you to assess diet quality from a sample of the faeces from grazing animals. So it's a pretty major innovation in that we now have an idea of the quality of the diet that animals that are grazing are consuming. So you can see here that both crude protein in blue and the digestibility of the diet in red, for a large part of the year, are at a very low levels. And in fact, if you look at 6% crude protein as about maintenance, see that for a large part of the year, the animals are consuming a below maintenance diet. So protein is a recognised biological constraint for Northern Australia for a large proportion of the year. And low protein leads to low energy intake, which means low production. This can be addressed using urea to some extent, either fed in a fairly basic trough system like this, or a more sophisticated urea into drinking water system. But this only increases growth enough to ensure survival of the animal, not to really increase production or reduce the age at slaughter. So one of the things we started doing fairly early in the piece in our research was to look at establish some response curves to the major supplement types. And we've sort of lumped these into the protein meals, things like cottonseed, copra, palm kernel, etc. And the so-called energy sources like the grains, barley, sorghum, and also molasses, and various combinations of those. And a lot of this work was done in pen trials in various places where we had control over what the animals were eating and we could measure their intake, and we also had a measurement of their performance. And the sort of thing we ended up with was response curves like these, where we have along the horizontal axis the intake and the vertical axis the growth performance of those animals. And in this case, this is just an example. We're comparing cottonseed meal in blue and the barley in red, and looking at young animals, about 200 kilograms live weight, probably about 8 months of age. The dashed lines here, and older animals, probably about 430 kilograms, the solid lines. But I'm not too worried about them. Just that we're trying to establish the shape of these response curves over a fairly wide intake range so that we can use that data for various feeding scenarios. And we can broadly lump all of that into response curves, group them into response curves like this, where we've got a range of experiments. and take it one step further and put them into generic boxes, as it were, looking at the energy sources and the protein meals. Now, there's a lot of variation around those, and I'm not pretending that they're representative of all different grains and different protein meals. But they do give us an idea of the shape of the curve and what we can expect. And the area in particular that we're really interested in is at these low intakes when we're on the fairly steep part of the curve, which is where the cost-effective supplementation is most likely to occur. One of the practical outcomes of this sort of work is that we can put together tools like this one we've developed, which we call WhichSup, which is just a decision support tools for producers and their advisors, where they can put in a growth rate, a growth response that they're looking for, put in the cost of the supplements as landed on their own property, and just do a first cut look at what the cost in cents per day would be to get that particular response, or probably more importantly, the cents per kilogram additional gain, to give them a feel about whether they're likely to get an economic response or not. One of the problems with supplementation is shown in this slide. These are the main grain growing areas within the red lines. The blue shows the main cotton growing areas, which produces cottonseed meal and whole cottonseed as supplements. And then in the green is the main sugarcane growing areas along the coast. There's a bit of sugarcane in the north as well. But what you can see is that most of these potential supplements are grown in the coastal regions or in the southern regions, a long way away from these more extensive production systems. So the cost of supplements, especially in the last decade or so, has continued to increase, and the freight costs increases that even more, and they've increased much more quickly than the prices that we have. So one of the things Dennis will talk about is the possibility of growing a protein source on farm to overcome some of those limitations. One of the other things we've looked at is how we can use a lot of this data. Now, we can't look at every type of supplement and develop a response curve. There'd be thousands of combinations, but we should be able to predict them using our own feeding standards. And these are sort of the Bibles of an animal nutritionist, as it were, in that they encompass the information that we have about the responses to known nutrient intake. And each developed country has their own, they're all basically on the same basis, looking at energy and protein, mineral intake, and the production that one might expect from that. But the problem is they don't always work in your own situation. They provide a rigorous framework, but for instance in Northern Australia we've found that they don't tend to predict in some circumstances very well the production that we're actually measuring. For instance, grass feed, which was developed by CSIRO and which is based on the Australian feeding standards, seems to predict quite well in the southern regions of Australia and the temperate zones, but not very well in Northern Australia and our tropical systems. In fact, some people go as far as to say the feeding standards don't work in the tropics, and therefore probably 2/3 of the beef production in the world have no system that they can rely on. So we used our own response data and all the information that we put together to look at this and test some of these feeding standards and see how well they did apply. So we used a lot of our own data with diet combinations and so forth. And we looked at two feeding standards in particular, the Australian feeding standards, the SCA, and the Cornell system based on a model that was developed at Cornell University in northern New York State. And what we found when we put all this data together was that they actually did a reasonable job of estimating the live weight change, so the predicted live weight change using the models, gave a reasonable estimate of what we measured in our pen feeding studies. So our conclusions were that cattle in the tropics aren't really different. We needn't throw the feeding standards out, that they were a reliable basis to use as a model, and therefore we could use them, but they need to be continually tested with new diets, new feeding systems, and particularly new genotypes and combinations. So the other practical question that was asked, as I said, we've developed a lot of this work as component research, and that's been a really useful tool. But the question's always asked, how do you put that into a commercial feeding situation? So when you've got a situation like this which goes across seasons, and the seasons vary in length and severity, where do you put your nutrients in? If you're putting them in the dry season, do you put them in early, soon after weaning, when the cattle are young, theoretically fairly responsive to additional nutrients? Or do you put them in later, closer to the end point, when the animals are older? Or is it a combination of both? And do both groups need the same type of nutrients, and do they respond in a similar way? So we've just completed a major study in Northern Australia, a grazing study, where we looked at some of these practical problems and issues. This graph here shows along the x-axis, once again, time or the age of the animals, with live weight on the vertical axis. And we've followed these animals through from weaning. This treatment here is just a typical low input system, fairly typical of much of Northern Australia, a urea-based supplement in the first year, and then no further supplementation after that. And you can see, as I described before, this typical sawtooth pattern of production. So the animals were eventually killed out here at 3.5 years of age with a carcass weight of around 300 kilograms. At the other extreme, we had a treatment where we put lots of inputs in, high inputs of supplements in both the first and second dry seasons post-weaning. And these animals were killed a year earlier, 2.5 years of age. The supplements we used were just sort of the commercial, what's often called MUP, which is molasses, urea, in our case copramyl, some minerals and some rumensin. And this was fed ad libitum in both years, shown here. And the intake was about 3.5 kilograms there and about 6 kilograms for the older animals. In between those two treatments, we had another treatment where we only fed in the second year, so high input supplements just in 1 year as a comparison. Once again, the same supplement and the same ad libitum intake. And then we broke that nexus and we looked at an improved pasture treatment. In this case, we used Lequina grass, and the animals were trucked down to a pasture at brine pastures. And they were on that for about 8 months and gained 200 kilograms at a fairly high rate. And they also were killed at a younger age, about 2.2 years. This is some of the cattle here on the dry season Lequina, not much leaf left there. And then in the wet season, and there's more leaf. This is all the treatments put together, so you can see the relativity, and you can see that these treatments were really reducing the age at which we're killing the animals. I'm not going to go into much about the slaughter data or anything, but I just want to make 3 main points about this. Well, one thing that I should say is that we had HGPs, hormonal growth promotants, superimposed across all of these. And I'll make one only point about that, in that the HGPs are still the most economical treatment that producers can apply in terms of increasing growth rates. One of the points I'd like to make is around compensatory growth. Now, this is a phenomenon where if you restrict the growth of an animal during a period and then you remove that restriction later on, the animal tends to grow faster when that restriction's removed than for a cohort group that didn't have that restriction or had a lesser restriction. And this is demonstrated here. For instance, at the end of this dry season feeding period, the advantage to the red group was about 47 kilograms. At the end of the dry season, The cattle then just grazed native pasture during the wet season, and that advantage was reduced to 29 kilograms through compensatory growth by these groups. Second year, we increased that advantage through feeding out to 107 kilograms, and in the following wet season that advantage was reduced to 74. So the compensatory growth has a big effect in reducing the response to supplements. And overall, in 2 drafts that we ran and 4 dry seasons, was to the effect of about 30% of the advantage we had was reduced. And of course that has big implications when you're going into a feeding program. The other point I'd make here is that the blue group here that was fed in only 1 dry season actually caught up to the red group that was fed in both dry seasons. And that was at a cost of about $60 less. So it demonstrated a very important concept, and that is that you can get to the same endpoint through quite different growth paths and with different financial inputs. And that's something that can be exploited through building in some of these efficiencies. This is just some of the cattle that weren't supplemented in that second year as opposed to those that were. The third and final point I'd make is we're interested in the skeletal growth of these animals. The reason we're interested in skeletal growth is the skeleton is what the muscles are hung from. So the more skeletal growth we get, the more muscle growth, the more meat produced. And we didn't really have much information about this when we started this study. So we measured the skeletal growth by measuring the height of the animals. Gives an indication of the increase in the growth of the long bones of the animals right throughout every month or so. So this just shows 2 of the treatments, the low plane treatment and the high plane treatment. This is the live weight data I've already discussed. And up here we've got the change in height. So the one point I really want to make about this is while these animals were losing weight during the dry season, they were still gaining in height at a relatively high rate, about 30 millimetres per 100 days. So the animals were pouring nutrition into the skeletal growth over and above muscle and fat deposition. And we think this has implications in terms of increasing or exploiting that compensatory growth effect. And so that's led to some of the work that's being done up at QUASP at the moment. So at this point I'm going to hand over to Dennis.
Okay, so in the skeletal growth part, Stuart has been talking about how animals are growing, and we're working on this hypothesis that the skeleton itself is the key driver for the growth of the animal, and that compensatory growth is one of the issues that we want to investigate. So what are we trying to do practically here? If we could keep that skeleton growing at an even faster rate, at the higher levels of nutrition, we end up with an animal of a higher frame size at the end of that dry season who's framed for this compensatory growth. And compensatory growth is one of the easiest ways in which we can increase the efficiency of the production system around, particularly in relation to the cost structure, as Stu pointed out. So we're looking at these different frame size type animals. So the animals that do not get much of a supplement or any at all over the dry season versus those that do. And you can see there's quite a marked change in their frame size. So what does that actually look like? The key hypothesis is that skeletal dimensions and elongation dictate the growth of the animal. Now there's actually quite a lot of work around this in relation to chickens and rat models in terms of how growth actually proceeds. And in fact, even in today's university news, I see Mike Waters and them have been talking about growth hormone affecting skeletal growth in humans and how this provides a new target for manipulation of individuals. So there is a lot of kind of general theory around about that, but probably less so within the animal sector. The sort of things we're looking for, as Stu indicated, forget the nature of the experiment. We've got young animals which are going to be growing through to bigger. They've got a large amount of frame size and their growth will actually be higher both in terms of weight and height. And then we've got, of course, mature animals where the skeletal growth is actually slowing down as they approach maturity. And think about yourselves going through puberty and approaching maturity, even though humans are a little bit different to cattle in terms of when the growth plates actually close over. So this led to an experiment or a project where Lisa Kidd is the CI on this, looking at the skeletal elongation in animals. And we've designed a series of experiments, and some of them which we've piggybacked on other work, looking at how nutrition itself affects skeletal growth. So in the nature of nutrition, we've had diets of low ME intake and low crude protein content, allied with diets which are very high ME intake and high crude protein content, to look at the differences that occur. Interestingly, we've got a growth hormone experiment starting in about, well, as soon as McLennan can get his act together and get us cattle, starting in about June or July, where Lisa and a PhD student, Risa Antari from Indonesia, will be looking at the effects of growth hormone on skeletal growth and development. Once again, we're using this standard scenario of diets. I'll explain a little bit later, but essentially they are crude protein deficient diets, high protein adequate diets where energy is restricted, and diets which have got high energy and high protein intakes. We've also piggybacked that onto P deficiency experiments also funded by MLA, looking at growth of animals. And in all of these, we've been trying to link these to some of the gene expression and metabolic pathways associated with metabolism. So on that particular point, I just wanted to point out that Stuart and I have been doing the same old work for 25 years, as everybody keeps telling us, and that's true. You can do quite a lot of these applied experiments looking at growth responses, and it's no different to these sort of agronomic experiments you might perhaps do in the plant area as well. Nevertheless, that data put together, and if you have in place where you want to go with that data, they provide the very functional model, in this case a functional animal model, by which you can look at some of the underlying mechanisms. Too often we've got some sort of technique or whatever, and we're trying to generate something in a laboratory that's going to create those differences. Well, we've taken the other approach of generating those differences out in the field and then trying to go back into that to see how those differences come about. So that means you do need very carefully controlled experiments, either in pens or in grazing experiments, and that's been the advantage of the work that Stu was just pointing out, where there's a large database there now that we can interrogate for a whole variety of reasons. In this work, if we're going to look at the skeleton, we have to look at what's actually happening at the tissue level, at the growth plate. So I don't know if you know how bones grow, but we have our long bones, of which we're going to be dictating most of the muscle mass, and that these have growth plates associated at either end. And we can sample into those particular bone structures to look at what's actually happening. So in the general scheme, it's a bit like a skyscraper going up, where you've got a cell division layer, and then you've got an elongation or hypertrophy layer, if you like, within that bone. But you can sample those bones, and Lisa Kidd and Simon are here sampling, I think that's at COSP, sampling into the tubercocci, so one of the hip bones, and that's a representative and showing you how the bone is actually growing. And as an example of that, and we won't, I think the details aren't all that important really for the purpose of this talk, but nevertheless, we have both Brahman and Holstein Friesian animals going through our 3 different diet types that we talked before: ad lib lucin, a high energy protein diet; restricted lucin, a low energy high protein diet; and ad lib Mitchell grass, a low energy, low crude protein based diet. And when we do the biopsies, and this is all Lisa's work, so all the work I'm presenting is somebody else's work, not mine. In her biopsy, she's able to therefore to detect the proliferative zone and the hypertrophic zone in the yellow there, which are the 2 areas I'm talking to you about how the bone actually grows. And so she measured, well, because she's got height here, but it's really width. It's the height of the width of those particular tissue types under histology. And so the key thing here is that there are treatment differences emerging between breeds and between diet types in the nature of that proliferative zone. And similarly, if we do the same thing and looking at the hypertrophic zone, once again we get treatment and genotype effects emerging there as well. So I think just provides an interesting story about how we're now getting down to how these results are coming about at a more basic tissue level, if you like. one of the advantages of things like biopsy is that you're able to go back into these sample types and do experiments that would have been more difficult to do in the old days. In the old days, when we were trying to look at metabolic pathways, whatever, we'd be having to take tissue slices or doing some detailed radioactive analysis or whatever, and those things could not be done out in the field. Now with biopsy types, we can do those things out in the field and come back and use gene expression type analyses to examine what's actually happening. So here are the, Simon and Lisa working at Brunchilli, which is just north of Tennant Creek. And I think actually where's Stephen? I saw him here. Yeah, Stephen's been up in these areas also collecting. So the other part of the work, which I'm not talking about today, is the endocrinology side, which Stephen Anderson is doing, looking at some of those control mechanisms as well. But you can have very good models, and these animals are on phosphorus deficient country, getting cows and growing animals, and so you're able to look at what's actually happening with those groups of animals, where there are quite large differences in many cases, and find at a more detailed level what the kind of functional mechanisms are behind that. And I think that's a real advantage in what we're trying to do these days. So in conclusion, the compensatory growth is a really interesting problem that still impacts on production systems. And looking at the skeletal growth side provides us with insights into how that's coming about. And whilst the histology side and the endocrinology side and the gene expression side doesn't necessarily lead us to a solution, immediately, at the first instance at least, it's a greater understanding of what might be open to manipulation. And I think that's the interesting thing that's coming out of this work. And it's very early days, so we really haven't got much data other than that preliminary stuff I just showed you before. The second area is about the growth paths and proteins. Stu mentioned that crude protein is the first limiting nutrient, and that's fine. You know, we could change around the production systems in Northern Australia easily, like this, overnight, if we could put a protein meal into those animals. And all those animals would reach all these high-priced markets, and we would be able to fix up the supply chain aspects and everything. The trouble is, all the protein supplements are too expensive. All the fish guys and the chicken guys and the poultry guys and the pig guys dictate and take away the protein supplements. So the ruminant people are not able to use those, just too expensive. So the problem is not the biology, the problem is the cost. So we're looking at, is there a way that we can develop a cheaper protein source on farm? And so algae is one of these, and Pierre is here somewhere, and he's been involved in developing this part of the work. So algae, we know, is a novel protein source. It's used in a lot of industries. There will be byproducts coming out of those industries that we can use. The trouble is they'll also be costed at the same way the current protein meals are, and the fish people and the pig people and the poultry people will get first bite at it, and from a ruminant point of view, it won't solve our problem. So we have to actually try and grow this material on farm in a cheaper way or a more robust way for us. To show you some of the results, this is Tander Penjaitan's work for his PhD, and he was looking at microbial protein production in response to spirulina. And it just shows you that algae work extremely well, and no different to any other protein meal supplement, in affecting microbial protein production. But more importantly, also affected the efficiency of microbial protein production. So both stimulated intake as well as the efficiency of microbial protein production. And the importance for that is microbes are the major protein source for a ruminant animal. About, on average, 70-odd% of the protein that a ruminant animal absorbs comes from the rumen microbes. If we look at the growth rate of these animals on algae, so this is work from Giogo Costa's work and Stu's up at Bryan Pastures, looking at the average daily gain against the supplement intake, we got exactly basically the same response of algae and cottonseed meal. So it works. So this led to the MLA project that Pierre is heading up, and it was all about screening microalgae for growth, determining some of the water and fertiliser requirements for the algae, and to develop simple ponds and harvesting methods. And Pierre's all about setting up the simple pond-based systems, and this is just showing you going through the multiplication stage of the algae, going in through the raceways that are out there at Pinjarra Hills and various harvesting, settling scenarios. So this is really all about trying to set robust systems up for growing algae on farm that will generate the protein meals that we need. We have done a calculation originally, and now it will be less because his yields are higher, but previously we've calculated something like 2 to 2.5 hectares of surface pond area would generate enough protein meal to feed 1,000 weaners over the dry season. So it's not out. It's within the realms of practical delivery, and we just need to develop more robust systems to do that. So in conclusions for that, finding solutions to crude protein deficit in the dry season is still the most important strategy or problem that we are facing. There are novel solutions, and algae is only one. There are many others that we ought to put our minds to. The problem is the cost and robustness of the system, and we've already got some strains which are growing quite rapidly. The final thing I want to finish up on is using these procedures to try and look at some of the underlying mechanisms and metabolic pathways, and how we might identify how these nutrients are affecting these things is of interest to us. So if we're going to look at growth, we need to understand skeletal, muscle and fat growth. So we need very good animal models. And those growth paths that Stu showed you in some of those pen studies provide those particular models. And by the use of biopsy samples in those and a combination of histology and gene expression, we're able to build up a picture of how those animals are proceeding. So I haven't talked about like the muscle. There are differences in muscle fibre type, for example. There's a whole range of scenarios like that as well. The one example I will talk about briefly today is the one about phosphorus. And now this was headed up by Simon Quigley, looking at how phosphorus affected growth of animals. So these were looking at the response of animals to phosphorus content. So here we have the live weight, a pretty standard experiment, different levels of phosphorus in the diet, and you can see that the animals grew at quite different rates. So that at the end of phase 1, which was the end of the growing part under the different diets, there were quite marked differences in weight of those animals. Phase 2 was when we added a phosphorus supplement, and of course they all started growing again. But this was set up as a very simple experiment to look at the effects of phosphorus and to look at some parameters about phosphorus adequacy and how we might identify those in the field. It had nothing really to do with what was happening metabolically. But nevertheless, with the appropriate sampler types, you can start to pull that sort of information out. We're interested in the height of the animals. Did they grow? And Stu mentioned before that animals in the dry season grow slower, but still grow. And similarly here, the animals on the low phosphorus diet were still growing. Interestingly, that's about the sort of growth we get in the dry season, 25 millimetres, 100 days. And in the wet season, we get somewhere between 60 and 70 millimetres per 100 days. So in phase 1, you can see these differences in growth rate between different phosphorus content diets. This is what the animals look like, and you can see that the only thing different between these two animals and the diet they got was the level of phosphorus in the diet, because it was a formulated, pelleted diet, everything else being equal. And you can see the marked effect that it actually has on the animals. Similarly, if you look at the height of those animals, you can see that there are differences in the height as well. So by taking samples at these various points, we can do comparisons along a growth curve, comparisons between low and high phosphorus, comparisons after supplementation, a lot of different sorts of scenarios. And this is what Simon has actually done, and he's done this as I indicated to you before, some of the endocrinology side of this Stephen's been doing, but we're not talking about that. So in conclusion then, we've progressed from examining responses to supplements, we've worked through databases, we've been looking for novel food sources and trying to understand the mechanism of the response. But to do this, you need a very good animal model. And secondly, you need to have people around you who can apply these techniques to old problems, and so make two grumpy old men like Stu and I look pretty good, like we actually know something. So that's why we're talking to you today. So just to show you where we've come from, I think that's the last time Stu McLennan ever looked at a soil sample. And at that stage of our career, we were thinking about, should we do plant science or should we do animal science? So we chose the more challenging, intellectually stimulating path, and we did animal science. But nevertheless, given what Joe Hockey has said to us in the last few days, and for Robert and the people in the plant science and the food science and nutrition groups of QAFI, I think Stu and I are ready to step up, and we think we could contribute to those other divisions as much as we've contributed to the animal science division. So thank you all very much, and we'd particularly like to thank many colleagues and students, and I can't mention them all, of course, here, but we have had two main funding bodies, that Meat and Livestock Australia and Australian Centre for International Agricultural Research. Right, thank you very much.